you go. Join. Meeting ID. I don't know. So for some reason, plugged this in today, and it's just not picking up all the things that it's picked up every single time that I've done this in the past. It's like, yeah. So. Right. Now meeting here. Okay, like that. Open. Cool. Haha. Oh dear. I've got to cut that volume down.
You have to mute first.
Yeah. Okay. How are we now?
Just still unmute. Okay, just don't talk.
All right. I'm just going to see if I can mute this completely. No, oh, hang on a minute. Oh, no. No, I haven't got the headphones. Wrong ones. Oh, unless they're that.
Welcome, participants. We're just waiting for other joiners to join us, participants to join us. There were more than 100 who registered, so we're counting on having them with us also.
Perfect. Yeah. Yeah, we're not sharing it.
Okay, so maybe we'll just start in 3 minutes. There are still some participants coming in. Can you all hear us well? Sounds okay? Yeah, okay. So we'll start in a few more minutes.
Thanks.
Of course, we see some familiar names. Our Biotech Information Center partners and scientists in the Philippines and other countries. We have representing here from Thailand, Pakistan, and many others. So I think we have to start now. So while waiting, I'm just going to read to you some of the important things that we need to consider. So good morning, afternoon, evening, everyone. Welcome to the first Asia Oceania Regional Animal Biotech webinar series. This is hosted by the USDA and implemented by ISA Southeast Asia Center here in the Philippines. I am Dr. Dora Romero Alvinita. I'm the director of the ISA Southeast Asia Center and the Global Knowledge Center on Crop Biotechnology. And I'm a trained scientist on genetic engineering in the US and a postdoc in Germany. So we gratefully acknowledge the support of our ISA Biotechnology Information Center network, including Malaysia BIC, Chinese Society for Biotechnology, Indonesia Biotechnology Information Center, Nippon Biotechnology Information Center in Japan, Pakistan BIC in Karachi and the Lahore chapter, Vietnam Biotechnology Information Service, and our new addition, the Biotechnology Alliance Association of Thailand. So we have 7 webinars in a series, and this will be conducted monthly to feature science and opportunities for biotech animals, biotech covering genetic engineering and genome editing for food and agriculture. So we will be looking at the status in poultry, aquaculture, swine, and cattle, as well as discuss in detail the food safety, environmental safety, and some tips on effective communication of this technology. So each one of those is a webinar topic. So webinars and workshops are currently being conducted in Africa and Latin America with other organizers, including ICA, ISA Presenter, and the Virginia Tech USA. And going back to what I said a while ago, so we are aiming for 2 speakers per webinar. But this time, our webinar today, this will be on in Asia and Oceania because we have varied stages of animal biotechnology developments and regulatory readiness in the region. Thus, it is fitting to share the innovations taking place in the region, as well as examples from other parts of the world. So this will allow us thorough appreciation and acceptance of the technology to enhance food security and agriculture sustainability. So this is a 1 hour webinar featuring our scientists from CSIRO or the Commonwealth Scientific and Industrial Research Organization in Canberra. We're currently on live streaming at ISA FB and YouTube channel. So all of your microphones are on mute, and questions should be entered in the Q&A function. The chat function will be used by the panelists and for important messages to and from the participants. At the end of the webinar, we will provide a link to a survey and respondents will be given certificates. So let us now welcome our moderator, Dr. Mark Tizard, the team leader and principal research scientist at the Genome Engineering for Health and Biosecurity Unit of CSIRO, who will set the scope of this webinar and introduce our speaker. Dr. Tizard, please, you have the floor.
Thank you very much indeed, Ola. And thank you particularly to Ola, to ISA, and to the USDA Foreign Agriculture Service for being a driving force behind getting all of this going. And I know that Ola has put a lot of time into this series. And also a thanks to all of you who are taking the time across Asia to come and listen to these series of talks. And I also have to say, in respect to the traditional owners of the land on which we talk from here in Australia, we recognise their elders past and present. That's the Wadawurrung people in the area that we are in Geelong, which is just south of Melbourne, right down in the south, the cold and windy south of Australia at the moment. It's a place I call home, thanks to the person that I'm going to introduce to you. Tim Doran and I have worked together for over 2 decades now, and it was through connection with Tim that I got my first chance to come to Australia and embrace it, and embrace Oceania and the Asia region as a home. Tim has also shared with me a long history in developing aspects of molecular genetics and finding useful applications of those. And of course, the subject of this and forthcoming presentations is animal biotechnology. Tim basically grew and developed a group here more than 15, 16 years ago with growing expertise in the space of poultry molecular genetics to the point of being able to develop transgenic poultry and now gene-edited poultry. CSIRO has an I in the middle of its name, that's industry.
Oh.
We have a focus to bring science to industry and to purpose. And so it's with that in mind that Tim has tailored a talk to you today to give you a feel for some of the things that are going on in this space of poultry molecular genetics that will lead through to, we hope, applications in the field of agriculture. And that's the issue at hand here, animal biotechnology having a use in the real world, addressing problems that we're all going to face. So without further ado, I'm going to have to hand over my headsets here to Tim, and I will get his presentation up to share. So just let me know if we are now sharing appropriately. I'm hoping you can now see that. Yep, that looks like we're fine. So I'll hand over the headset to Tim and introduce his presentation. Here we go.
Well, hello, everybody. I hope you can hear me okay. And thank you, Ola and everyone for the invitation to present today. And thanks, Mark, for that very warm introduction. Yeah, so I'm going to give a presentation today on the work that we've been doing in CSIRO in Australia. It's our Australian Research Government Organisation. So some work that we've been doing for quite a period of time now around developing genome engineering specifically for poultry species and predominantly in chickens, entitled Genome Engineering in Poultry Opportunities and Impacts. So I'm based at the Australian Animal Health Laboratory, which is a high containment facility based in Geelong, where we're giving this presentation from today. So there's a little bit of a flavour of this talk around the application of genome engineering for health impacts in poultry as well. Just clicked, Mark, and it didn't advance. Just bear with us while we get the technical. So just arrows. Okay, so first question is, why do we genetically engineer birds? There are a few reasons. One is, biology. So chickens, quail, in particular, in terms of poultry species, are really good models for studying biology, particularly developmental biology, and that's because we've got really good access to the embryos as they are growing and developing in the eggs. So we can study them, quite easily. So, we like to apply the latest and newest molecular tools to help us study developmental biology in birds. The main reason for our lab and our team being involved in genetically engineering birds is agriculture. So, we're interested in how the technology can be applied to improve poultry production. That's meat and eggs. So we're generating chickens that are resilient to diseases, that have improved production traits, and are improved in terms of the welfare outcomes for the animals that we're farming, and that the products that we harvest from them are safer. Biotechnology in general is another reason. So there are some groups globally that are using the chicken egg as a bioreactor for valuable biologicals. So that's something that our team isn't necessarily involved with, but it's another reason why other groups out there are trying to apply genetic engineering in chickens. Can we use the eggs as a biofactory to make proteins of high value? And the final one there is a growing area around conservation. So there's growing interest in being able to use these new tools to try and protect endangered bird species. So it's a concept called genetic rescue, and that there's a spectrum of work there that leads right through to some way out there kind of opportunities that include de-extinction, and maybe some of you on the line have heard about some of the goals of some of that work, which is around being able to use existing species as proxy for extinct species, and somehow trying to create living proxies for extinct animals. So, but our focus here really is primarily around agriculture opportunities. A little bit about how we do what we do. So we work with primordial germ cells, or PGCs. They're the stem cells in a chicken embryo that go on to become sperm or overproducing cells. And it's really interesting in birds, as opposed to many other animal species, these stem cells, primordial germ cells, migrate through the vasculature in an embryo on their way to establishing themselves in the gonad. So that means we can actually get at them. We can harvest them and work with them in a very early stage embryo. And there's kind of two ways of doing that. You can harvest these germ cells from the blood and work with them outside of the embryo. So it's a really very highly skilled technology where you can culture the cells in the lab, introduce genetic modifications into those cells in vitro, expand the cells into clonal populations, and then inject them back into recipient embryos to generate germline modified birds. Or you can work with the cells, we say, inside the embryo. And this is a technology that our team here in CSIRO developed, where we showed that you could directly transfect those germ cells with genome engineering tools while they're still circulating through the blood. We call it direct injection. And there are lots of reasons why this technology is actually beneficial for our lab as a way of being able to modify the germline of bird species, and particularly chickens. And you can see there's an image there of sort of the famous GFP, green fluorescent protein chickens that we've generated, and many other groups now globally have generated birds that have that rather spectacular phenotype as a tool to show that the technology is working. So a little bit about this technology that we call direct injection. So we developed it several years ago now. I'm looking at that publication. So it was published in 2013. So we have a paper that describes the method. So the tools that we use in this method are either transposons to insert a transgene into the germline of birds, or gene editing tools, and CRISPR is the main one that we work with to be able to edit DNA sequences in the germline, either by deleting genes or making slight modifications to targeted regions within the DNA. We simply inject those tools in a transfection agent into embryos at a very early stage of development. So it's called stage 14, Hamburger Hamilton, but essentially that's at day 2.5 of embryogenesis. So we take a fertilized egg, we incubate it, we take it out of the incubator at day 2.5, we open it up, and you can see hopefully down on the bottom right hand side of that screen, unless there's a pointer that I can use. Mark will try and get a point. Can you see the cursor as I do that? Oh, here we go. Brilliant. Yeah, so this is what the embryo looks like at day 2.5. Here's a glass capillary that has our genome engineering tools in a transfection agent, and we just simply inject that into the bloodstream, transform the germ cells while they're circulating in the bloodstream at this stage. Once we've done the injection, we seal the egg back up. We've created a little hole to get access to the embryo. We put that back into the incubator. And then once that's sealed and incubated normally, we take those chicks, those embryos through the hatch, and we grow up those chicks to sexual maturity, and we identify the next generation that we can then use in a breeding program to generate efficient germline integrations or deletions in their offspring through breeding. And we end up with about 1 to 10% of the offspring having that introduced genome modification. So it works quite well. So the next slide is just to really give you a feel for the different types of applications that we're using this technology for. I'm not going to talk about all of them today. I'm going to focus around being able to use the technology to improve resilience to disease in chickens in the agricultural system. I want to tell you a little bit about a project that we have to try and improve the safety of eggs, and that's our allergen-free egg project. And I'm going to sort of finish off with talk to you a little bit about a project that we called Vaccimizer, which is about improving eggs for vaccine production. So a little bit of a background on that project before I get to it, and that is that fertilized eggs are used a lot in the production of many vaccines, including flu vaccines. So we've been doing some work to use genome engineering to improve eggs for that process, and I'll take you through a little bit more about how the science actually works with that example. But you know, we are also using the technology for a range of other applications. I believe Mark's already had a chance to brief this audience around the work we're doing to solve the issue of sex selection in the egg layer industry. And we're also, you know, we have lots of collaborations and happy to work with groups that are doing more sort of functional genomics, where they use the chicken as a model organism. So we've been doing some work with groups that, you know, studying how organisms develop and grow in embryogenesis. But the focus for us and the focus of this talk is really around applications in agriculture. So I'll focus on disease resilience to start with. So this isn't all our work. There's quite a few groups globally now that have an interest in applying genome engineering technology in the poultry industry. And there are three diseases that most of the work is focused on. And the first one is around avian influenza. And so there are a couple of ways that you can apply genome engineering to disease resilience. You can either use comparative genomics to identify differences between organisms that have high levels of resistance compared to organisms that are susceptible to disease, and then try and introduce those differences into the susceptible species. Or you can use a synthetic biology approach and introduce something brand new into the genome of an organism that would not normally be present to make it resistant. And I'm going to give you a couple of examples of those. So around avian influenza, I know you'll all most likely be aware that it's a major problem in the global poultry industry. There's consistent and extensive outbreaks, you know, pretty much all the time, and particularly in recent years. Controls difficult, vaccinations is not always fully effective. And then, you know, I guess topically in our current time of COVID pandemic, you know, there's always a threat that an avian influenza virus will spread, you know, from poultry into humans and potentially cause a pandemic as well. So there's that kind of human aspect to why working with avian influenza is important. So there's some work going on at the moment around a gene called ANP32A, and it's a really important gene in all organisms that are susceptible to influenza infection, and it's a host protein that interacts with the viral polymerase. And what the group led by Wendy Barclay in the United Kingdom identified was that there are species-specific differences in ANP32A protein that results in a host restriction. It sort of explains why some viruses only grow in certain species and why others don't. And so what she was able to identify is that there's a difference in this ANP32A gene. So there's a region within the chicken gene that encodes for a 33 amino acid insertion, and that difference is enough to, if you can remove that and use gene editing to remove that insertion, so that you now have a more humanized version of that ANP32A gene, then avian influenza viruses don't grow in the avian cells that have that deletion. And Wendy and her team, in collaboration with the Rosalind Institute in the UK, were able to make this deletion, remove that 33 amino acid insertion, and show that that avian influenza viruses were unable to grow and replicate in cells that had that deletion. So it's an example of where we can use comparative genomics, look for differences between why viruses can grow in a certain species and not in another, and then now replicate that using genome engineering in an organism like chickens and make them resilient to infection. So it's a very exciting target. There's still quite some work to do to take this through to a demonstrated application, but there's a lot of work going on, and I sort of flag that as an example of something to keep an eye on, and I'm sure that we're going to see some really exciting and exceptional work that's going to come out of a number of groups globally around targeting this gene to try and improve resilience to avian flu in chickens. The second one, again, is around a comparative genomics study, and this is with another important virus of poultry, avian leucosis virus, and in particular subgroup J, so ALVJ virus. So it causes significant economic losses in the poultry industry globally. The receptor for that virus in chickens and in other species that are infected by avian leucosis viruses is a gene called NHE1. And scientists were able to show that if you had a deletion or substitution of one single amino acid within the protein that gene encodes, it can confer resistance to infection. And it may be a little bit difficult to see this alignment on this slide, but what it shows is that the chicken and the red jungle fowl, from which our modern day chicken has evolved from, all have this tryptophan residue present at position 38 within the sequence. But other related poultry species like the guinea fowl and the quail don't have that conserved sequence. And that's what makes them resistant to infection by ALVJ virus. So chickens, turkeys have it, they're susceptible, the other species don't have it. And so another group were able to show that if you could, in the chicken, precisely edit that region within the NHE1 gene so that you no longer have tryptophan present at position 38, it renders the chickens completely resistant to avian leucosis virus. And this was a really very nice study of how absolutely targeted specific gene editing can result in a really beneficial phenotype within poultry. And I think that this is the best example at the moment of how genome engineering is likely to be taken forward around disease resilience in the poultry industry. The final one is a synthetic biology example of some work that we've done here in our lab. So this is work that we've recently been involved in, and it's around another important pathogen of poultry, Marek's disease, so the Marek's disease virus. So our idea was based around introducing something brand new into the chicken genome that wouldn't normally be there. And it's actually very much related to the genome editing field, and that is by introducing the CRISPR system that is normally found in a bacterial cell and confers resistance to viruses that infect bacteria, called phage. So basically transplanting that system across into the chicken cell and setting it up in a way so that it targets the Marek's virus, which is a DNA virus, and the CRISPR-Cas system is a nuclease system that's targeted towards DNA. So we were asking the question, can we introduce a brand new system into the chicken cell that specifically targets the DNA of the Marek's virus when it's infecting the chicken cell, and then get targeted cleavage disruption of that virus so that it can't replicate? And it worked really well. And so this is a paper here on the right-hand side of the slide that maybe you can have a look at, that talks about that research, how we generated the transgenic chickens, took that through to in vivo challenge experiments with those chickens, and showed that we could get high levels of resilience to the infection in chickens that contained this Cas9 protein system that we've essentially hijacked from a bacterial cell. So there's 3 examples of work that's currently going on at the moment to develop chickens that have levels of resistance to viruses. And there are some others that I haven't had time to talk about today, but it's a really growing area of research globally. And one of the reasons is that the poultry industry have done very well over years just using conventional selective breeding technologies to improve productivity. But disease resistance is a trait that's very, very difficult to see improvements with just conventional selection. So, the industry are very much looking at new tools like genome engineering tools to be able to help find ways to be able to improve the animal's resilience to important diseases caused by viruses. Okay. The next example that I wanted to talk about was around increased food safety, and this is particularly around egg allergy. So egg allergy is widespread. It impacts more than 40 million children worldwide. It's predominantly an allergy of children, and many children grow out of the allergy before they reach adolescence and adulthood. It costs, just in the USA alone, up to 25 billion per year. So it's a significant food safety issue for the global poultry industry. Egg allergy is caused by 4 proteins that are present in egg white. So egg white's made up of about 40 proteins altogether, but 4 of them can cause allergy. They are the ovomucoid protein, ovotransferrin, ovalbumin, and lysozyme. The predominant protein that causes allergy is ovomucoid, or OVM. It's the most allergenic. We know it makes up a small amount of the total egg white protein, and it's resistant to cooking and digestion. So of the 4 proteins that cause allergy, 3 of them are actually denatured by baking and cooking, so they're then rendered safe in egg products. But OVM resists those processes, so it's always there, and causes allergy even in cooked products. So there's a group in Japan that published this paper that again is on the right hand side of the slide, that showed that you can use gene editing tools to delete this gene from the genome of chickens and still get a functional egg. So our work that we're doing is saying, okay, right, you can take that egg product now and use it in cooked and baked food products that use eggs that will now be safe and not allergenic. So we're currently using CRISPR to knock out the OVM gene, using the capability that I've talked about. We know it'll be safe in cooked egg products. We're hoping that this will be the first gene-edited animal food product on the market in Australia. We're doing lots of engagement and interactions with the regulators here in Australia to ensure that that's the case, and that's a very valuable and fruitful interaction and relationship that we have at the moment that Mark is very much at the centre of. And we're really hoping to achieve something truly novel and disruptive in the very, very rapidly growing free from food industry. Globally, there are a whole lot of free from products, whether it be gluten-free or allergen-free foods that are being developed globally. So we're looking to use gene editing to create some egg products that are free of this major food safety concern around allergy. So that's one I wanted to flag as well. Now, the final one is a bit more sciencey. So I'm going to sort of take you through how we actually get the technology to work in a practical sense. And this is the project I alluded to earlier around next generation of vaccine eggs. So we're developing high yield eggs to enhance vaccine production. So as I mentioned before, the main focus here is around eggs for flu vaccine production. So currently it takes flu vaccines predominantly are grown in fertilized chicken eggs. Here we go. And currently it takes about 2 eggs just to make 1 dose of flu vaccine, so 1 shot, so 2 fertilized eggs. And so, our goal is to try and improve that and try to get, a 10-fold increase in the amount of vaccine you can get per egg. So our goal is, maybe with just 1 egg, you can end up with, 5 shots of vaccine. So the big flu vaccine manufacturers rely on fertilized eggs. The major companies use lots of fertilized eggs every day that they're manufacturing vaccine. So for some of them, it's greater than 1 million eggs per day. And then that creates issues for them. You know, at the end of the day, they've got greater than 1 million fertilized eggs that are now infectious that they have to handle and treat as waste. So they're very interested in being able to use a lot less eggs to make enough vaccine for us all. So our goal was to use genome editing to identify inhibitors of vaccine growth in the egg. So we know that there are genes that respond differently to influenza virus growth within an embryo. So we used a genome screen. So we use small interfering RNAs to target a whole range of genes that are being expressed in the embryo when we infect them with the virus, and identified genes that cause the virus yield to go up or to go down. And so obviously for us, we were looking at the ones that make the virus yield go up. And we identified several gene targets. We validated them a little bit more in embryos using, again, another RNA interference approach using short hairpin RNAs, and then went on to generate a target gene knockouts using gene editing tools. So the gene that was at the very top of our list after we did that screen was a pretty obvious candidate. It's a interferon alpha receptor gene. And so it's not surprising that you know, interferons are inhibiting vaccine virus growth in eggs. You know, they're part of the normal immune response to viruses in chickens and also in embryos. So we targeted to delete that gene from these chickens and see what impact that has on the growth of the viral vaccine strains in eggs. So the gene itself is located on chromosome 1 in chickens. Here it is here in red. It has a whole range of different exons that are important to express the mature protein. So our goal was to essentially delete the entire first exon of this gene using CRISPR-Cas9. So we identified 2 guide RNAs which are used to target the Cas9 nuclease to that region of the genome, and cut in 2 places and remove a segment of about 100 to 150 nucleotides that remove that exon from the gene. So to make that work in practice using our direct injection technology that I spoke about, what we did was to develop a plasmid that expresses the Cas9 nuclease plus those 2 guide RNAs that target that nuclease to the interferon alpha receptor gene. We showed that that vector was able to express the Cas9 and the guides in a chicken cell line. We knew it was working. So then we get in 120 eggs that are fertilized for the direct injection process with this single plasmid that has the CRISPR genome engineering tools encoded on it. So essentially we inject, and again, you've seen this image before on a previous slide, using a microcapillary directly into the dorsal aorta of this embryo at day 2.5, we inject 1 to 2 microliters of a complex of that plasmid with transfection reagents. We then seal up the egg. After the injection process of the 120 eggs, we were able to transfer 88 to our farm facility at day 14 of embryogenesis. And of those 88 eggs that we transferred, 28 hatched. So from 120 eggs that we injected, we end up with 28 chickens that hatched at the end. Of those, 12 are roosters or males, and we end up working with the males. And so what we do is we raise those roosters to sexual maturity. And once they've reached sexual maturity, we take a semen sample from them and we screen that sample for the presence of the deletion in their sperm cells within the semen sample. And we use a technique called PCR to amplify that specific region within the genome, and we're looking for the deletion. And where the red arrows are pointing to these are the PCR products run on a gel, and you can see that there are 2 sizes. The top band is the normal or the wild type interferon gene that's not obviously had the targeted deletion, but the smaller band that's present in these 2 samples is the deletion. So we know at this stage that the edit is successful. It's present in the sperm cells of these roosters that we've taken through to sexual maturity. And what we found was that of the 12 roosters that we kept, 8 had the deletion in at least one of their semen samples. And we take multiple semen samples just to be sure. We then take that PCR band that has the deletion and we sequence it and absolutely confirm that the targeted region that we want to delete has now been removed in those samples. So we know it's worked, and we know that it's in the germ line of these roosters. So now we need to breed them. So we're able to quantify which of those roosters which of their sperm cells have the majority with the deletion, so we can choose the best ones. So we took 3 of those, so chimeric rooster 1, 2, and 3, and we placed them in matings with 6 wild type hens each. And you can see here for rooster number 1, in those matings, we were able to hatch 480 chicks. So they're the next generation, so they're G1s. And 14 of those were positive for the deletion, and those deletions are now in the germline of those birds, so they're not chimeric anymore. Every single cell has that deletion in it. So that was a transmission rate of about 3%. For rooster 2, kind of similar results. We hatched 340 G1s, 5 were positive, transmission rate of 1.4. Chimeric rooster 3, we ceased that mating. We didn't have a positive at the time that we ceased it, but we generated enough positive G1 germline edited chicks by that stage to move forward. So again, from that overall experiment, we were able to generate 19 positive G1s from close to 1,000 chicks that we hatched at a transmission rate of about 2.1%. So it's really similar to what we had seen previously using our direct injection technology to make transgenic birds. So for us to make a gene edited bird, it was very similar transmission rates. So the next step for us is to just screen that product, that deletion product in those G1s, just to make sure that it's what we expect it to be. And of course it was, and the deletion was very consistent across all the G1s that we screened the deletion in, which is really good to know, right? Because it shows to us that the CRISPR-Cas system is a very reliable, very specific tool for being able to make these types of deletions. It worked very well in our hands. Then the next role is to mate, so that, obviously they have the 2 copies of this interferon alpha receptor gene, one from the male and one from the female parent. And so at this stage in the G1 progeny, they only contain 1 deleted copy. So they're heterozygous. So we have to mate 2 heterozygotes together to end up with offspring that have both genes knocked out, and they're the ones that we need to take forward for this study. So we did the breeding of the G2 generation to generate homozygous birds that have the deletion in the IFNA gene. And that was successful. And then we did a little bit of phenotype characterization just to show that the deletion of the IFNA gene in progeny that have both copies removed is working. So we essentially take cells from wild type controls, heterozygotes and homozygotes, stimulate them with MX gene expression, or stimulate them with interferon alpha and then measure MX gene expression, just to show that they're responding as you would expect for a gene that has, a chicken that has the gene deletion. And we were able to show that there was no interferon alpha receptor 1 activity. So that was, it was really good. It confirmed that we were able to generate these gene edited birds and generate homozygote progeny. And then we obviously then took that forward to say, now do eggs from these line of chickens that have the gene knocked out perform better for vaccine production? And the answer is yes. So here we have 4 different types of influenza vaccine viruses that are used in seasonal vaccine production that are grown in eggs that have come from our interferon alpha receptor knockout line. And you can see that in all 4 examples, we're getting higher levels of virus in the knockout line compared to the control. So this was, and so we've got improved vaccine production characteristics in these eggs. So this was really exciting for us, and just wanted to use that as an example to show you how the technology works in practice, how we go from being able to use our direct injection technology, identify a gene target, make a specific deletion in that gene, and take it through to an impact. So just this final slide is just to show how the poultry industry works. And so we've got close links with the global poultry breeders. And the way the poultry industry is set up globally is that there are only a few major companies that breed the majority of the world's commercial poultry. So it's quite possible that you can work with one of those companies and then have major impact in terms of being able to get your the change, the modification that you're introducing into their birds, reaching out into the global industry. So you can, it's a very good species to be working with to have impact around, I think, genome engineering applications for industry. At the end of the presentation, I may have gone a little bit over time, so I apologise if that's the case. But just finally, just like to thank the team that we work with here in the lab. I won't go and name everyone individually, but we're really lucky to have a great team of researchers that work with us on these projects. And finally, just like to thank Mark, who's front and centre here in this picture, for giving me the opportunity to talk today, and for leading the way in the interactions with the work that he's doing with you all online. So with that, I'll say thank you and happy to answer any questions, and hope it's been an interesting talk.
Well, I can see there's a couple of questions that have come up, so I'll take you through those questions. And in the meantime, I'd just like to say that, Tim, that picture of the group you saw is a group of people that Tim has built up over many years, both through promoting the ideas, getting the capability and the funding and resources to pursue those ideas. Obviously, we want to take this into the real world, and that's where I've had a chance to play a part in the team and actually start to interface with regulators and groups like yourself who are looking at bringing this forward. Now, we'll hold the microphone like this to make sure that we can get the answers between Tim and I. So the first question is, what the consumers and the government's response or impression to gene editing in livestock products? So I'll pass to Tim, and I can add some comments possibly.
Yes, so you can hear me okay. We've got an interesting situation going with the headphones here. It's a really good question. So, we've spent a lot of time working on what we think are applications that are more likely to get consumer acceptance to their adoption. So, for example, being able to use the technology to improve health and welfare of the birds that we're farming is much more likely, and we know is a better way to get consumer acceptance around adoption of the technology. So we believe that we are working on applications that are likely to get consumer acceptance and adoption. But we know we could also use the technology for other applications that are likely not to get consumer acceptance as well. So around welfare, health of the birds, disease resilience, and food safety, we think we're on the right track. It's similar with the government's response. They're interested to see it used for the right applications. But here in Australia in particular, there's support from the government to develop and apply new technologies to animal agriculture. And so there's a general acceptance around the need for these types of tools to be able to provide, food security for our population and the world's population over the coming years. So generally, it's good.
Thanks, Tim. That's a really good coverage of the answer. I'd say also that for CSIRO, we we look at things from end to end. So Tim has given you a really excellent run through of one end, which is some really smart ideas in the laboratory that face towards questions that the industry faces and would like to get solutions to. But of course, to make it into the real world takes a whole bunch of stages. So from one end, the lab, to the other end, which is in the real world, puts us across the path of many things, including the regulators and governments who oversee them, and ultimately the public. And I think Tim has described really nicely there that what we're aiming to do is to address specifically the consumer at the end point, but taking in mind what the regulators are going to consider about the safety of the product in the middle, and also something that faces to a problem for industry, because industry's got to want to adopt it to actually make it something that can go through ultimately to the consumer. So the second question is about the IFNAR chickens, the vaccine improvement, and it's asking the question whether the gene edit will impact the chickens, the ones producing the eggs, in terms of their disease susceptibility and survivability. That's a really good question. I'll hand over to Tim in a second, but I'll just also say the Q&A is open, so please throw in some questions, or else we'll just be talking about things we want to. I'll hand to Tim to talk about that issue of disease susceptibility in the IFNAR chickens.
Yeah, that is a really good question, and something from the outset we were obviously alert to. Obviously, if you're removing a gene that's normally there to limit virus growth or protect or be part of a normal immune response, then you're likely to be generating chickens that are more vulnerable. And that is the case, but it's definitely manageable. So we certainly know that flocks that are kept specifically for production of eggs, for vaccine production, are kept in quite high biosecurity environments. So they're quite clean environments anyway. So that would need to be the case for our line of chickens as we take them forward with vaccine manufacturers. But also we were able to show that we can still vaccinate our flocks against, common disease threats that pose issues for layer lines. So they still mount a good immune response when vaccinated, so that we can still have a reasonably good health biosecurity program to protect these flocks from disease threats. But it's a good question, because without doubt they do pose more of an issue around their susceptibility, and it's something that needs to be managed. But overall, it can be managed, and the end outcome of having a less requirement on eggs for vaccine production and improved vaccine yield sort of outweighs that little issue.
Okay, at this stage, I'm not seeing any further questions. If you were to list one drawback of IFNAR 1, what would it be? So there is a new question. Let me throw back to Tim to see if he can answer that one.
Yeah, so I'm not sure I could list just one. I mean, so it's not the end of this work as well. So we've actually generated 2 different knockout lines that have improved uplift of vaccine growth, and we're now looking at others. So the way I would answer it is that we're now actually specifically looking at genes that are only expressed within the choreo and allantoic membrane of the embryo, and that's actually where the flu viruses grow in embryoted eggs. So it'd be much better to be able to target a gene that's only expressed in that membrane, not throughout the whole embryo, and obviously within a line of birds that we're using to generate those fertilized eggs. So I think moving forward, you'll see us working and refining the gene targets that we're working on. IFNAR works fine. The other gene target that we're working on works fine as well, but it would be much better to be looking at genes that are only expressed in that part of the embryonated egg, so that it doesn't have more of a widespread impact on the whole animal. I hope that helps answer your question.
I think that was actually a pretty good answer, and I'm seeing another question. This is great. So we've got some questions coming through now. Do you have any tests for gene-edited chickens to grow for growth and nutritional values?
Yeah, so that's an area that we haven't been involved with. So when I talked about consumer acceptance and industry need and adoption of the technology, the industry's done really well with conventional selective breeding around growth and food conversion and nutritional value using conventional technology. So we don't believe that that's the right target for this type of technology just yet. I think once we can see the industry adopt the technology for the types of applications that we've been talking about today around disease resilience, health and welfare, which are very hard to use selective breeding to see improvements for, then it's likely to drag other opportunities along with it, such as improved growth. We can identify some targets that would be useful to or could be used to target with gene editing now that would improve growth. Myostatin would be one gene that comes to mind, but at the moment we don't think it's the right application to have impact with this technology just yet.
That's a good answer, Tim. I like that. So the last we've run to the last few minutes here. I think that I'll take this as the last question and then hand back to Ola. It's when do you expect to see the full commercial release of GM Poultry that CSIRO is working on? And I would modify that slightly to say GM or gene edited poultry products. So Tim.
Yeah, so we are anticipating that some of the work we're doing now will be adopted by industry, most likely here in Australia first. And my best guess at the moment may be around the allergen-free egg product, single gene deletion to make a safer food product. But we're still a little way away from that being a reality. We still need to work with a commercial breeder to get that into their flocks and get the products out there. So it's not likely to happen for probably maybe 2 to 3 years, but we're kind of working on that time frame and we're hoping that we'll get there. And there are a couple of other projects that we're working on as well that are definitely working with industry with the goal of being able to see that work adopted as well. But that's for another time, I think, to talk about that work.
Brilliant. Thank you very much, Tim. We won't pester Tim with any more questions, but there's one last one, quite long, but it's actually very simple, and that is, what came first, the chicken or the egg? Something that's been bugging one of our listeners, and I would say we do have an answer for that. And in spite of, in the dictionary, the chicken comes before the egg. Well, we would say in evolutionary biology terms, the egg came first. As evolution changes, something hatched that was a chicken, but its mother wasn't quite, and she would have laid an egg from which that thing that now we would call a chicken was hatched. So the first thing was the egg, and from it emerged a chicken. So there we go. In the meantime, I'll let Tim go, and I'll hand over back to Ola to take charge.
Mark, can we also, there are still 2 questions in the chat box from Margaret and from myself.
Yes, let me see. Okay, yes. Well, very quickly then, before Tim does rush out the door, what have we got? So,
and it's not livestock. Have you done any public perception study on genome editing?
Oh, actually, I can let Tim go. And
okay.
yes, and the answer is yes.
My question is, okay. Right.
So just very quickly to that one, CSIRO, as we say, we work from end to end. And that is from the clever idea to the public. And we do have people within CSIRO that face directly to the public and have interactions with them, do surveys, questionnaires. Yeah, we want to know that the public is going to accept the things that we're doing. So that was that one. And Ola, your question.
On avian flu. Do we have
yes, and this was about the current problem with avian flu, correct?
Yes. Yes, right.
Yeah, so the answer to that one is no. We aren't specifically working on vaccines for disease in poultry. There are big companies and other research groups that work on vaccines. We're working at the molecular level with the bird itself, and as Tim described, to try and change it so that the flu virus can't get hold and can't replicate in the bird. and that's kind of a molecular vaccination, but not one you can wheel out and give. You have to grow the birds and then distribute those. So it's a longer process, but we believe ultimately a better one.
It will come out better. Okay,
Yep.
so we'll let you go. But before that, I would like to thank, let's give a big round of applause, virtual applause to Mark Tizard
To
and
Tim.
of course to
To
Tim
Tim particularly.
Doran for all of this hard work that you have provided us. It's very interesting. Actually, maybe this will be the first time for some people like me to hear a lot of information on the biotechnology in poultry. That's why we made it the first
Yes.
the first topic in the series. So
And we
thank
were
you
very
so much.
grateful for that. It was great to have the chance. And yes, Tim, I think hit it just right to have why and then some examples of exactly how we do it, because it is kind of nitty gritty, but it's great to have the chance to speak to this audience across the whole of Asia.
Yes.
So thank you very much, Ola, for organizing.
Okay. So with that, we thank you all. We thank also the participants who were able to register and come in. So we would like to again say that biotechnology in poultry is for the biology itself, for agriculture, for biotechnology, and for conservation of genetic resource. So we would like to invite again everyone to please respond to the. Have we posted
The survey.
the survey? Yes. Okay. So it is there. Please respond to the survey and we're going to provide you your certificates as soon as we have your response. And we would like to invite you again to our next webinar, which is going to be on aquaculture, and it's going to be in June. And we will have participants from, I think we'll have another one from India and from Japan, hopefully. So we will just be alerted with our social media in Facebook and in our Crop Biotech update for the link to register. So thank you so much. And this is going to be aired in YouTube. And of course, it's still in Facebook. For those who were not able to get in, please tell them that it's so interesting and you're going to learn a lot. So thank you so
Also, thank you
much.
to Godfrey for organising all the technicalities in the background, and to the other of your colleagues
Okay.
at ISA.
Yeah.
It's run
Yeah.
very smoothly from our side. Thank you.
Thank you so much. Okay, goodbye for now and see you once again. Thank you for for everything. Yeah.
Thanks,
Bye
everyone. Thanks
bye.
for coming and seeing.
Yeah, see you next week.
Bye.
The meeting. Bye bye.